think we're in business. All right. Whoa, okay. Hello. Hello. There's such a lag on here though. Right. Can you guys just let me know if you can hear sound? If the sound is good, please let me know because right now I do have a different screen, but I can't actually turn on the audio because there'd be a lot of delayed feedback. So if you can let me know in the text box, that would be awesome. Okay, thanks, Aaron. Thanks, Zane. Jeanette, thanks so much. Hey, Shannon, great to see you. Hey, Megan. Sophia, how are you? Mystica, how are you guys doing? Hi, Cam. All right, sounds like we have some good sound. We'll get going in a few minutes. I'm here to answer a lot of questions. I'm really here to try to make sure that there's less anxiety about what's coming up. We're pretty nervous ourselves, to be quite honest. I really want this class to go well and finish well, so we're going to try our best to make sure that everyone feels like they're going to feel accommodated. We're going to talk a little bit about that. That's why I had the text box open. So I am going to be monitoring that. There is about a 20-second lag from what I can see. So I will check back and forth, but just bear with me. And I'm hoping to kind of answer your questions that you might have today. And I'll be using that little live stream chat box on the side. So are you guys nervous at all about what's going on in the university? Also, apologies for not putting the lecture slides online immediately. Apologies on that. Feel free to. I'm hoping that you all are aware. Mic is making weird static. I'll try play that. Okay, I'm gonna just figure out what's happening with the mic, so we'll be right back. Okay, I just switched to a different mic, Max, so I'm not sure if that helps at all. I'm hoping it helps. I think it's a little bit better. We are using this mic for a different course, so. Okay. All right. Okay, we have switched mics. If you can just let me know if this mic, because we're switching back and forth, so if you can just let me know if you can hear me clearly, that would be awesome. Again, apologies on this. It's the world of online lecturing. All right, and I think we're getting pretty close to the time that we're supposed to start, so why don't we just go for it if you're all okay with that? All right, so I think we're going to just get started if that works. And yeah, of course this battery is now going to die on me, but we'll hope for the best. Okay, hi everyone. Thanks for joining us today. So I have to say, and I have to admit, I kind of miss being in class, and I hope that all of you are doing really well. I hope that you have been keeping healthy. I hope that your families have been keeping healthy. I hope that you know that everyone here at the university has your best interests at heart, and we're trying our best to make sure that you feel supported. No, I think we're good with the microphone now, so I think we're good. Um, so I just wanted to mention that we're going to go through a couple of different things today. I'd love to hear your feedback as much as possible, and we are going to use the chat box and the live chat. So if you can let me know like what your thoughts are, and if you have any concerns, please feel free at any time to do so. It's really all about you. These courses are for you, and I really do miss having you in class, to be quite honest. So I hope all of you are doing really well, and I hope that you are taking really good care of your health. Just a reminder that you don't have any tutorials. I'm going to be posting that survey for you tomorrow, and I want you to make sure that you're aware of what's going on. And one of the proposals that we currently have, and I'd love to hear your thoughts on this, is what I've posted on the announcements page related to being able to just reweigh a little bit of the final take-home assignment if we go that route, just to give everyone 100% right now, immediately, if you decide to go with that vote. In terms of your participation, I know some of you are very anxious about that, especially in light of not being in tutorials. So we're hoping to just allay some of your fears and some of your worries and anxieties. We're not here to try to take marks away from you. We want you to continue doing as well as you have been up until this point in time. And this disruption is really just a terrible thing for many of you who were expecting to do well, and we want you to finish well, to be honest. The take-home will be 4 short answer questions. It will be based on lectures. It's not an assignment. You don't have to do any literature review. You will be given some practice ahead of time, so we will have some practice questions for you. Today's lecture will be the last official lecture that you need to worry about for your term test. And regardless of whether you do an assignment or whether you do the take-home assignment, the format will be the same. It will be all the lectures after term test number 2, and it will also be just up to today, whatever we get to finish today. And if we don't get to finish, I may add a little bit, or I may decide that that's enough for you to be able to answer the final assignment or final exam questions well. Does that sound okay to everyone? Could you let me know how you're feeling about that, or if you can tell me if there are other things that were on your mind or that you're anxious about? So Megan, I think the biggest difference for me is that if we were to go and do the final exam online, and it would have to be fully online, it would be timed. You would be given 2 hours, and with the take-home, the short answers, which would be similar format anyways, I would have the expectation that you would be able to finish within about 2 hours, but you get the full 48. I know it's stressful, and I know that some of you are looking at that as, wow, maybe it's going to be harder. It is not harder, but we also just want you, for those that have accessibility concerns, maybe you have multiple exams, just having 48 hours, it does a lot to help allay some of those anxieties. So the reweighing part, some of you are concerned that the final assignment might not be in your favor. And so if you don't do as well on the final assignment, then we would take a little bit less and we would include your term test and make that a little bit higher. You already have that mark available for you, so we want you to be able to maximize your mark in this course. And I don't know if that makes sense to you or not. The marking criteria, if I understand it correctly, I'm not marking harder, Felicia. I want you to work alone because we are going to ask you to submit through Quarkus. We're not going to ask you to submit it through Turnitin, but you know, your TAs know you well enough, and if you've been copy pasting, people will know. Just be honest about it, but there's no difference in criteria. So Zane, in terms of like what the mark breakdown would be, so the advantages, like the main changes would be that if you don't do as well on the final take-home assignment, it would be worth less. So instead of 35%, it would only be worth 30%. And your first term test would be worth a little bit more. My expectation is everyone will do well on the final take-home, but it's to kind of reduce that anxiety. In addition to that, if we go this route, because I want you to know as many of your marks ahead of time as possible, we would immediately give everyone 100% on the participation portion in the class. I don't know if that answers your question or not, Zane. I hope it does. Yes, you would get an automatic, everyone would get an automatic 10 out of 10. I know you're anxious about that, and I know that you might feel that we're going to be marking you harder. We just want you to know that we're not going to be doing that. Do you have any other questions right now or anything that I can answer for you? If you're anxious, we do have like a discussion page that is up there. The survey will be available for all of you tomorrow, and we want you to be able to engage with that discussions page. We are also going to have an example in the next live session whenever that happens, most likely next Tuesday, to make sure that you all know what the expectations are. And regardless of whether it is a final take-home exam or if it's a final take-home assignment, which is, again, flexible in terms of the weighting, we are going to have a practice to make sure that you know like what we want on the short answers. The 2-hour final exam would be the same as we've been indicating so far. So it would be multiple choice. It would be one-line responses. It would be timed. You would only have 2 hours max. Again, the format would be very similar to, we might increase the number of multiple choice test questions. We don't really know yet how that would work, Jeanette, because we haven't heard from the Dean of the Faculty. So right now we're still learning a lot about how the online format would actually work. So it would be automatic. We want the reweighing to be automatic, Alexandra. So anyone, we want to tell you you don't need to ask for it. We would like look at your grades overall in the course for your term test, your final exam, and based on which one is higher and which one would give you the better mark, we would automatically reweigh that for you. We hope that by reducing the impact, if you didn't do as well on the final, or increasing that, we have to take a vote because we would be doing a reweigh, and that's the reason why we're doing this. Today's lecture material would indeed be on the final, but today's would be the last. I'm hoping to finish the most important parts of it for everyone, and today's material would be the last. You need time to work on your presentations. You need a little bit of time to also start thinking about your other courses and your other assignments. So regardless of whether it's the take-home assignment or the take-home final exam, it would still be the same number of questions. It would be 4 short answer questions. I'm not sure if that answered your question, and sorry about the delay. Thanks for answering that, Aaron. Yes, 4 short answer questions. There are lecture slides up there now, Jessica, and apologies on that. I had thought that I had checked off one of the boxes, but I had not. Will I still be doing a distilled slideshow as a review? I will probably be doing that, yes. Okay, I'm not up to date on the emojis, so I'm not sure what those little things mean, but okay. Cassandra, I think if I understand your question, the final assignment would be available for you on the 9th, and then you would have 48 hours to be able to answer that. So yes, that would be correct. Boy, it's really weird watching me here and here, so. I would assume that so I'm answering Grace's question, I would assume that it would be just based on the short answer questions. If you want additional short answer questions, if that makes you feel better, I'm happy to include like one-liners, multiple choice. It would be basically an open book. If that's up to you. If you want, we can include those, or we can have them optional for the take-home assignment. If you wanted to do them or have them included as your mark, we could do that. So everything's kind of gone really silent. So are you guys doing okay? Are there any questions that I've missed in the chat feed? Is this still going? I'm hoping. So I'm just going to type this one because I think it's faster. Yeah, I think Tabina, that's correct. Sorry, this is like a little bit weird, kind of going back and forth. So apologies if you watch this later, it's going to be a little bit disjointed. Option 1, midterm 25, final assignment. Yes, that if you didn't do as well, it's going to be an automatic regrade. So Omar, the actual options are we keep everything the same and it's an online final exam, or you have that flexibility that's automatically guaranteed to you. That's the vote that you're taking. We keep the current marking scheme and evaluation. We just go ahead as we planned right from the beginning, and you get a 2-hour, no aids allowed type of final exam that's all done online. I don't know how they're going to do that, but it will be delivered online. Or you get to do a take-home exam. We adjust your participation immediately right now, and you have that flexibility option. It's not option 1 or 2 that you're voting on. You're voting on keeping things the same, or having that flexibility automatically guaranteed for you. I'm hoping that's clear. I'm going to wait about 20 seconds because that's how long it takes. If you can indicate whether that choice is clear, that'd be awesome. This is so weird watching me over here. So I've also been getting a lot of questions about the tutorial assignment. So Grace, just to reiterate, so if you go the route of having the final assignment, the major advantage is that you would have 48 hours to think about the questions versus the online. It would be like a 2-hour regular but done on your computer type of final exam. So that choice is yours. I'm not trying to sway you either way. And it would be very similar in format. That's right. I'm not trying to make it harder. And that's why I'm emphasizing it. I'm not sure if that's clear or not. And again, please feel free to follow up with any questions that you have online as well on the discussions page if it's not clear. I would really invite you to ask those questions. That's correct. So the major thing, Emma, right now is that the 10% is automatically given to you because you're not in tutorials. It would be unfair to try to give you like different marks based on like very little interaction with your TA. You're not going to be there to answer any questions, so it'd be hard to give you that mark. So the rationale behind giving everyone 10 out of 10 as is would just be based on that. I am likely also for the tutorial assignment, there are about 10 students that have already submitted it. I would be pretty open and lenient about that as well. I also just wanted to check in. How many of you are okay with recording online for like a PowerPoint or finding alternative ways of delivering your group presentations? Are you guys all doing okay with that? Okay, so it sounds like most of you are doing fairly well with the recording, so that's great. Would it make you feel any better? And I don't want to take anything away from the 10 students that have already done the reflection. Are you guys okay with the timing for the reflection? I promise that we're marking very fairly leniently, like the average is going to be like incredibly high for that, so you don't need to worry that you're going to not get at least an A. Most of the ones that I've seen are pretty close to perfect or perfect. And I'm hoping that you'll just spend like 30 minutes or so doing that. So are you guys all doing okay? So Neha, yes, the reflection piece would be due on the last day of class. In fact, if we go the route of the take-home, we could probably make it due around the same time as you're submitting the take-home. I'm going to be pretty flexible on any of the due dates, to be quite honest, as long as you let me know. Right, so I'm hoping most of you are feeling okay. I'm hoping most of you are feeling a little bit less anxious now, if that's good for you. Maybe I'll spend about 30 minutes just going through the last sets of slides that you need to worry a little bit about, or not worry, but to kind of focus a little bit on. And throughout what I'm going to be talking to you about, I will try to emphasize what you need to know, potentially for that short take-home question or for the final exam. Aaron, I think it's the least that we can do. Yep. So Aaron, I completely agree. This has been pretty stressful for everyone involved, and I really worry a lot about how you guys are all managing and how students are managing in general. We don't get to see you, so we're worried about your mental health, your well-being, your finances, how you're managing all these different courses. So we do tend to worry, and it is stressful for us as well. So thank you for those comments. All right, so I'm glad that you're saying hello to each other. If we're okay, so can I just get on with some of the other lectures, or do you have other questions? I'm happy to answer them. Okay. All right. Lots of friendly people in this class. That's great. Okay. Since everyone is seeming to be a little bit more laid back and you kind of all know what's going on, so I think what I'm going to try to do next is just to go through some slides, if that works. Okay, awesome. So I'm just going to go through these slides now, and again, apologies. I'm not going to be able to look at the screen over here on my right immediately, but if you do have any questions, I'll pause and I'll try to make sure that I answer them. So I am going to emphasize that if you are doing the take-home assignment, it will be open book. You'll have like your notes. You can use your notes. I expect you to use your notes. And one of the model systems that we were looking at is whether or not we could use this INVI mouse model, which has chronic inflammation as well as papillomas, which kind of resemble skin cancer that results from wounding the skin. And we looked at in the last lecture how having this constitutively activated MAP kinase signaling pathway, this MEK1 pathway, could actually result in skin cancers in this INVI mouse model. And we focused a little bit on the INVI mouse model as well, and we were kind of looking at this constitutively activated MEK1 system, and that means that because it's using involrucin, and because this is only found within the skin, that as a result, within skin cells and keratinocytes within the skin, we would have constitutively activated MAP kinase, which is also known as MEK1. And that was what we had sort of talked about before this whole COVID-19 thing really took over our lives in the last lecture. And I'm hoping that that sounds a little bit familiar to you. This mouse model, as I indicated before the break, or before we had to take a break, is going to be on one of your short answer questions. And I am going to really encourage you to start thinking about it. And we will, in the next class or next online class, actually go through some of the pathways related to it. So we wanted to use this mouse model, a mouse model where if you chronically damage the skin by creating a wound on the back of the mouse, that their skin would actually develop these cancerous-like growths, which we call papillomas. And we wanted to know what is going on if we have these keratinocytes that kind of spontaneously seem to undergo proliferation. What is the link with chronic inflammation, the wounding and the tissue damage, and how it tends to overgrow? Can bacteria and responses to bacteria that we know are growing on the skin and the immune system kind of lead to all of these things? It's a relatively new model system, but it's one that we wanted to think about. And you need to think about this, again, for your final take-home assignment. So one of the things that we had looked at is that if we had somehow managed to remove the TNFR receptor in these INVI mice, and we had removed the TNF receptor specifically in the immune cells or the white blood cells in these INVI mice. And again, just as a reminder, these INVI mice, they're the ones that develop these tumors. So you put a little wound on their back, and they start to grow malignant, and they form these papillomas. But if we had eliminated the TNFR receptor from the leukocytes of these mice, we actually saw that they don't actually develop these tumors. And that's, again, something that we talked about in the last lecture. And one of the things that we saw was that in these mice that had this TNFR or TNF receptor ablation, we actually saw fewer of these immune cells within the skin. And normally in these INVI mice, we saw more immune cells without any damage or anything else in the skin And so these TNF receptor knockout animals that were crossed with the in v, so they have the potential to form tumors, but they don't have TNF receptors on their immune cells, they don't tend to infiltrate into the area of the lesion. And some of the different types of immune cells included these gamma delta T cells that we sort of looked at very briefly as well. So there's a selective reduction in many of these different types of T cells in these animals that potentially have the capacity to form these tumors. So these in v mice have that papilloma capacity, but there's also less of them if we also take out the TNF receptors on those white blood cells. And that's one of the things that we wanted to examine. And I sort of stopped on this last slide just before we went to break. And one of the things that we know is that when we look at how TNF receptors, what the activation is, and some of the pathways that lead to their activation, we know that many toll-like receptors that respond to different types of immunogens or different types of pathogenic factors, for example, toll-like receptor 6 or toll-like receptor 1 usually responds to bacterial lipoproteins, whereas TLR2, toll-like receptor 2, or TLRX will respond to peptidoglycans. And most interestingly for us, when we look at TLR5, toll-like receptor 5, and we look at flagellin, flagellin is usually the activator for toll-like receptor 5. Now, why is all of this important and why is this pathway important? When we look at how TNF receptors normally signal, we often see that they will activate this mid-88 or mid-MyD88 or mid-88, depending on who's doing the lecturing. And we know that this will activate the NF-kappa B pathway, and this results in some of those inflammatory cytokines. And could this be sort of a link with the TNF receptor response as well as the bacteria, because all of these are sort of bacterially related, except for this one viral component. And so could this be the reason why the lack of the TNF receptors are protective against the development of skin cancers in the form of this melanoma? And so the next step that these authors decided to do is there a link between bacteria on the skin of these mice, because they do have that, this TLR receptor signaling pathway, the generation of inflammatory cytokines like TNF alpha, because if we don't have the receptors for TNF, we don't get the inflammatory response. Is there a link between skin bacteria, skin inflammation, cytokine activation, and the reduction in the infiltration of those immune cells? So they decided to look at that, and again, there are a lot of different slides here, and I want us to focus on the two that I've outlined for you in blue, so in panel A as well as in panel D. And here, if we take a look, if we look at the normal formation of papillomas, and we are looking at these different in v mice, and these in v mice, we know if you wound the back of their skin, they will develop these papillomas. And here you can see that there's this really robust response that occurs about 2 weeks after wounding. They start developing these papillomas. And if we knocked out that mid-88 pathway in these in v mice that normally have these papillomas, do we still form those papillomas? And you can see in the knockout mice where we have knocked out this mid-88, we actually see a much lower response, suggesting that this mid-88 or myeloid differentiation primary response gene 88, which is normally the signaling adapter for a lot of the inflammatory pathway, if we knock that pathway out, we will blunt the papilloma response. We don't get those skin papillomas like we normally would. So the other thing that these authors decided to look at was, okay, out of all of those different types of toll-like receptors, and they go through all of these, so they go through them in a stepwise fashion, the only one, when they looked at mice that lacked toll-like receptor 5, that showed a reduced papilloma response. So when they looked at the toll-like receptor 5 knockout mice in these NV background knockouts, the toll-like receptor 5 seemed to be like an important mediator. So we have two of those different steps. So we have the toll-like receptor 5. If we knocked out toll-like receptor 5, we reduce the incidence and the size of these papillomas, reduce skin cancer, in other words, in these NV mice. And we also know that the MYD88, the downstream signaling for toll-like receptor 5s, are also important. So both of these things suggest that there is indeed a link using this NV mouse model with bacteria, inflammation, and the development of skin cancer, again, in this form as the papillomas on the y-axis. I hope that's okay for everyone. I know I went back and I covered that fairly quickly. I think, or I hope that that was clear for you. I'm going to tell you that you do need to know about the different controls and the different things that you might use to help analyze how inflammation would play a role. And next, I'm going to cover how they know that it is indeed the bacteria that play a role in all of this. And in your answers, if you were asked to elucidate this pathway, you would have to indicate these different types of experiments. Okay, so I'm just going to go ahead just for the sake of time, because again, I don't want to spend too much time going over all of this, and we do still have more slides to be able to cover, and there are a couple of other things that I want to cover here as well. So if we go back to that original slide, the TLR1, the TLR2, the only known ligand for TLR5, or the toll-like receptor 5, is known as flagellin. And flagellin is the main protein constituent of the bacterial flagelli. So they use that to help swim around. So the key question, the thing that we'd want to go back and to take a look at, so is it really the bacteria on the skin that somehow induce this skin cancer in the form of this papilloma? So how does it affect this wound-induced tumor incidence? So these mice that we're looking at, these NV mice, get treated with the broad-spectrum antibiotic, and I'm just going to pronounce it as ENR, and they were either given the antibiotic in the drinking water, or it was applied on their wound, so a topical application, to reduce the bacterial skin load. So they're reducing the overall amount of bacteria on the skin. And when they did that, when they used this ENR antibiotic, they saw that after wounding, the tumor formation was greatly reduced. The tumor size was also greatly reduced, so the numbers and the sizes of these tumors. Again, going back and in with the rest of the model, where we're looking at the toll-like receptor 5, we're looking at the MYD88 or the MyD88 pathways, and we're now looking at using antibiotics. All of this is suggestive that bacteria do indeed play a role in the incidence of these skin tumors that could potentially occur. In fact, the authors go one step further, and again, depending on what your final vote is, you may not have to start worrying about memorizing all of these details, because it would be more open book for you, and you'd have 48 hours to kind of flip back and forth and change your answers a little bit, or not have to worry about memorizing. But one of the things that was really interesting was that when they started to treat these animals with methicillin, which is a very narrow spectrum antibiotic, and it only targets gram-positive bacteria, and these are, again, highly abundant on the skin, and these would include things like flagella including species like Staphylococcus aureus. They actually didn't see an effect. So this suggests that only the types of bacteria that have flagelli are the ones that actually induce the form of skin cancer, because methicillin, which targets the gram-positive ones, which are not flagellated, seem to have no reduction in skin tumors. They form tumors just like they normally would. So all of this taken together suggests that bacteria that are living on our skin that will interact with the toll-like receptors on the immune cells and then activate the downstream MYD88 and cause the release of tumor necrosis factor, which would then recruit some of the other immune cells, are really important if you happen to have a mutation that will constitutively activate the MAP kinase or the MEK pathway in your keratinocytes, which is what we're looking at here. And so going back to what we looked at earlier on, is there indeed a link between skin wounding, chronic skin wounding, because we know that happens in individuals, and a link between proliferative responses and the immune system and specific activation of the immune system with the gram or bacteria that contain this flagellin molecule? There is indeed, at least according to this paper, a very, very strong link. So making all of those connections and using these different types of experiments and some of the negative controls like methicillin, which only works on gram-positive, non-flagellated, but very, very common types of skin bacteria, had no effect. So it's very, very specific types of bacteria that can actually induce all of this. So I'm going to pause here. There's a little bit of a lag. So I'm going to see if you have any questions or things that you want me to go back over. I'm not going to try to cover all of the things that you need to know for schizophrenia. And if I need to go back and tell you what you need to know, I will record that for you asynchronously. And then by the beginning of the next lecture, I'm going to also indicate specifically what you need to start studying, and we'll have a better idea of what you need to know for your take-home assignment or for your final online exam. Any questions? Could you please go back to the slide with slide 9 and methicillin? Yes. Okay. So let me read your question. So no reduction in tumor initiation was observed, methicillin. So there is tumor formation still going on in these mice. Oops. So they are still seeing tumor formation. Sorry. So Julie, to answer your question, you need to demonstrate that it's only specific types of bacteria. You need to link it back to the TLR5 pathway. So if it's just any type of bacteria, it might include like the different toll-like receptors. They had knocked out some of the other toll-like receptors that I did not cover in all of the different lectures, so or the slides. So we have all of these different types of toll-like receptors that interact with all sorts of different types of bacteria. The only one that seemed to have an effect was the toll-like receptor 5, meaning that they had to justify and fit the data to what they had seen with the toll-like receptor 5 knockout and the MYD88 pathway as well. Sorry, I'm kind of going back and forth, and apologies on this. It's just me in this room today, so I don't really have someone to kind of read off like what's happening, and there is a little bit of a delay, so apologies, because it's going to look terrible when you guys go back and review this later. Grace, to answer your question, I think what I'm going to probably do, based on the suggestion that was given earlier in the lecture stream, is to actually give you sort of a distilled version of all of the slides that you would need to know, regardless of what your choices are. We'll know by next week anyways. You guys will start the voting as of tomorrow, if that makes sense, and if that's okay with all of you. Oops. Okay, sorry, I'm getting a little bit caught up in this and trying to do both screens. If it's okay with you, I'm going to come back over some of the different chats that are going on here. I still have a few slides that I'd like to get through to go back and just finish off things. I'm probably not going to use the remaining hour or so to go through everything that you need to know related to the organoid system, and I'm probably going to not cover the organoid system, but you do need to know a few things about the schizophrenia pathway and why we might want to look at using a brain organoid, and it's probably going to take about another 25 to 30 minutes. Is that okay with everyone? If it is okay, can you give me like a thumbs up, or can you just indicate that the next 25 minutes or so will be okay? And that's all you'll need to know for your final exam. All right, so what I'm going to try to do is to take you through the last part. I will tell you that you probably need to understand some of the basic mechanisms underlying schizophrenia and why we might use things like organoids that are shown here to kind of help us to understand it. There's still a lot about the disorder, as you might remember, that we still don't really understand. And I'm going to go back and do a little bit of a review of some of the models around schizophrenia and how we might study or make use of some of the genetics behind schizophrenia to build a model system that we would be ethically able to use to understand what might be going on. And hopefully by the end of this section, over the next 20 minutes or so, and that's all we're going to do, so 20 minutes, just hold on, and if you need to get up, take a break, please feel free to do so. How we might use schizophrenia, which is a very, very complex disorder, to actually use brain organoids to study complex disorders of the brain and other systems like this one. So in the last lecture series, before we went to skin and before we started looking at in-V models and everything else, we started looking at how dopamine and other neural circuits might actually play a role within schizophrenia. And one of the things that we looked at was that dopamine helped to facilitate the learning of association. So we learned, for example, that often we see increased dopamine right before a reward, or we might see a decrease in dopamine activity and dopamine release if there is no reward. And so we know that dopamine signals that some things are good, some things are bad. And that was sort of the main hallmark of this. So we know that there's a problem with dopamine and dopamine signaling in schizophrenia, and we know that because when we start using different types of dopamine receptor antagonists, we can usually see some kind of effect on schizophrenia. And we talked a little bit about the different types of drugs that have been in use, like haloperidol and blockade of dopamine responsiveness earlier on. So we know that in schizophrenia, there's just a little bit too much dopamine, and as a result of having more dopamine around, that this will change the perception. And most of these are slides that I've received from my colleague in physiology, and she's really an expert on all of this. And this increase in the abnormal release of dopamine at inappropriate times might mean that if you suddenly started associating novelty and the release of dopamine, you might start thinking that something that is normal and what would normally be considered to be average might actually be more important. And this is why individuals that have schizophrenia often have different types of ideas that people are out to get them, or that they're feeling like they're being chased, etc. They just might see a stranger that they've never seen before, and they might assume that that stranger, because they're wearing the same color jacket, and that jacket takes on more importance because of more dopamine, they might make this really strange association because they just have too much dopamine going around. And this will lead to the creation of a cognitive scheme or a delusion to explain why everyone who is wearing a blue colored jacket, because you've associated that with one stranger, might be following you, and if every time you see that blue jacket, that stranger is out to get you. And again, that seems bizarre, but that might be the thought processes that go on in someone that has a delusional disorder that's common in schizophrenia. We also know that schizophrenia is a very complex disorder, not only because they gain delusions, and that's the positive symptom, but they also have negative symptoms. So unlike depression, where things just go down and down and down, it's all in one direction, we have an increase in things that we never had before, like delusional disorders, and we also have a decrease in cognitive functions. So we know, for example, if we were to look at the brain scans of individuals normally at rest, so these are normal individuals, and the brighter the colors are, and you can see the heat maps here, on this vertical axis, the brighter colors mean that there's more activity. And this is the front of the brain, and this is the back of the brain. We can see that at rest, normal individuals have a lot of activity in the prefrontal cortex, both on the left side and the right side. And we know that even at rest, schizophrenic individuals, if we were to measure their brain scan activities, we would actually see a reduction in activity in the frontal lobe. So again, very little yellow, very little red, unlike we see here in normal individuals. Now, when we ask someone, a normal individual or someone with schizophrenia, to do what is known as a cognitive task, and it could be a card sorting task, and it might be something that looks like this. So if you saw this up on a screen while you were in an MRI machine and you were asked, so out of all of these cards here at the top, choose the one that would have the same number of objects that the card shown here at the bottom would have. And most of you would think about it, and you'd be able to pick the ones with 2 stars because it would match the ones with these 2 crosses down here at the bottom. And you might make that even more complicated by saying, okay, showing you the same slide, choose now the card that has the same color as the slide here at the bottom. And again, most of you would have no problems being able to show that it would be this card here because it has the same color as the one with the 2 crosses. In a schizophrenic individual, because their frontal lobe is not as active and it's not working as well as it should, we actually see in this card sorting task very, very little activity. And in fact, if you were to examine it carefully, even though they're trying to figure out this problem, the prefrontal cortex, the activity tends to go down a little bit. And so we know that they have these negative symptoms. So they have an increase in delusional symptoms, and they can't really think cognitively as well as they should. In addition to that, on top of all of that, in these individuals, they also seem to have these cortical abnormalities. They also have changes in the levels of these gray matter volumes as determined by magnetic resonance imaging or structural imaging, even in individuals that haven't been treated with any type of antipsychotic drugs. So these are treatment naive, so they haven't been on a drug yet. It's their first episode, so they come in, they have been diagnosed with schizophrenia. They already, even though it hasn't been going on for very long, have these gray matter volume abnormalities. The cell density is much lower. The dendrites, the branches of neurons are lower. They have lower spines, which I'll show you on the next slide. When we examine the brains of schizophrenic, schizoid type individuals afterwards. And because this prefrontal cortex is a late maturing region, and because of the emergence of these prodromal symptoms in the teenage years, and because there is gray matter volume abnormalities, particularly within the frontal cortex, so the dorsolateral prefrontal cortex has a lower volume, and we know that in postmortem individuals it's also lower. And because they have problems in this area, as I showed you in the previous slide, with being able to think normally, that all of these things might be related together. Histopathologically, if we were to take a look at the top of a normal prefrontal cortex neuron, and what we're looking at here at the top is one dendrite, and here is another dendrite, and each of these tiny little protrusions that come off that look a little bit like mushrooms that you're seeing here at the top are what are known as spines. And what we're looking at here is a normal subject. And then down here at the bottom, you can see the the dendrite with very, very little spines. And we normally need these spines to be able to make synaptic connections. So again, this makes this whole model of schizophrenia quite complicated. So we see a reduction in activity, we see gray matter reductions in volume, so something's going on in the prefrontal cortex. When we go back and we stain the prefrontal cortex and neurons that are in the dorsolateral prefrontal cortex, we see that not only is there less gray matter, but the gray matter that's there also has structural changes that we can see when we do a type of stain which we call Golgi impregnation. So it's a type of silver stain that's used for neurons. So all of this suggests that there is a very, very complex disorder. So what accounts for a reduction in gray matter volume? What accounts for the lack of dendrites and spines, which means that there's something wrong with these neurons? And how do we also account for having like too much dopamine around? So one of these things that we need to be able to do in whatever sort of model that we are able to build is that we have to account for all of these signs and symptoms that we have just been summarizing or revisiting over the last few slides. So there is an abnormality in schizophrenia that reduces their cognitive frontal lobe activity, and we call this hypofrontality. They're not able to think really well. It's a negative symptom, reduced cognitive capacity. But how do we connect this also with the dopaminergic system? And again, in science, we need to be able to have a way to link all of the different observations that we see, and we need to be able to do that in a logical way that makes some sense for putting together all of the data that is being accumulated. So one potential model for all of this that might account for it, and it's just one model. There are many, many different models that have been suggested, but this one model does tend to make use and can incorporate all of the different changes that we've just been talking about, is that if we look at the brain as a circuit, there is actually a circuit going back to these dopaminergic neurons, and we can detect that in the fully developed adult brain. So this prefrontal cortex, the neurons that are part of the prefrontal cortex, is a way to control the dopamine neurons that I'm going to show you in a moment. So there's that link between prefrontal cortex and dopamine. And what this might mean, and it doesn't show you how this happens, but it does link all of the different parts and the elements of what might be happening, is that the prefrontal cortex does not mature properly and it's underdeveloped. And we know that in schizophrenic individuals that they have hypofrontality, they have reduced gray matter volume. That would mean that if we don't have enough neurons in the prefrontal cortex, or we just don't have enough signals coming from those neurons, we would have less feedback control on these dopamine neurons, and so the hypofrontality might actually lead to uncontrolled dopamine since we have less feedback control, and that means that there's more release of dopamine, and this might account for some of the positive symptoms that we see and we have been talking about. And this is known as the developmental or glutamate hypothesis model for schizophrenia. And the model system, which again you may need to memorize or maybe you won't, and you might have to do this as an open book sort of model system, but this glutamate model kind of goes along these lines here. So here is this circuit, and this is a circuit. It is a closed circuit. And it goes back and discusses all of the things that I mentioned to you a little bit earlier on. So Dana, I just took a look at the side panel here, and I think a lot of that will depend in terms of the study guides. We're a little bit behind on the study guides, and so at the very least, I'm going to go back and provide for all of you a distilled version of all of the key important slides, regardless of take home or doing a final online exam. But in terms of the study guides, I think we're going to wait and see what the result of the vote is, because on one hand, it might help more for an online final exam, getting you ready for multiple choice, one-line responses. And if you're only doing short answers with like an open book, it might be less necessary. I hope that makes sense to you. So here's this model, just going back, and it is one that you would need to know regardless, is that from the prefrontal cortex, we have these neurons, and here's the neuronal cell body. Here is sort of the axons, the white matter tracts that go back, and they would make connections to an area of the brain known as the ventral tegmental area, or the VTA. And within the VTA, the neurons actually are dopaminergic. And these dopaminergic neurons that you see here, in turn, kind of release dopamine back onto the prefrontal cortex. And normally there is a tight regulation. So this would be used to turn off this pathway. And as a result, there'd be less release of dopamine from this mesocortical pathway. And when it's necessary, it would turn itself back on. And one of the key things to connect this with the next slide is this is the normal sort of connectivity. But what might happen is that if there is a pathway that is not as robust and it's not as effective as what is being outlined here, then if this pathway is not working well, then what would happen is this pathway here on the left would actually become overactivated. And that's what we see. We see hypofrontality, less activation or less overall activity, which also means there would be less inhibitory feedback. And that would mean we have more dopamine, which would fit the model system that we looked at before. And it turns out that one of the key things, the prefrontal cortex releases a neurotransmitter known as glutamate. So this is the glutamate model. The prefrontal cortical neurons release glutamate, and normally that responds to this pathway being activated. And so these NMDA receptors that are found in this pathway seem to be important in the feedback control. And I'm going to show you that in the next slide. So there's one other group of neurons that's important in this, and you have to connect what's on this slide with what's on this other slide that you see over here. So one of the things, again, to keep in mind, we have the prefrontal cortical system, and I've shown you on 2 slides earlier on today that as part of this hypofrontality that we see a reduction in the gray matter volume, and we see a change in the types of neurons that we see within the cortex, that when we look at this feedback inhibitory system, that the cortex normally would release glutamate, and glutamate will bind to these interneurons, and it's not shown to you here, but these interneurons that are here within the area of the brain known as the ventral tegmental area, and apologies to keep flipping back and forth, this ventral tegmental area, or VTA, the interneurons are the ones that have those NMDA receptors. And these NMDA receptors, in return, that are found on these interneurons normally are there in the VTA and also within the substantia nigra, but we're not worried about the substantia nigra right now. And when these get activated by glutamate, these NMDA receptors cause the release of GABA, and as a result of the GABA release, they will turn off or inhibit the dopaminergic neurons within the ventral tegmental area. So here is that normal feedback loop. Now we know that in the schizophrenic individuals that there is a reduction in this pathway. And so the reduction in this pathway leads to a lower or less activation, a reduction in these inhibitory neurons being able to do their job. And if they're not doing their job and turning off dopaminergic neurons, that means that this pathway as a result is going to be increased. We would expect to see much more dopaminergic release. So hypofrontality, less activation here, would result in more activation over here on the left. And again, I hope that makes a little bit of sense to you. And one of the things that we want to do is to understand this model system. We know that it's a fairly good model system. It's not the only one that's out there, but it's a fairly good model system, because if we use a drug known as ketamine, and ketamine is a drug that normally, when taken, can block the NMDA receptor, not just the inhibitory interneurons, but all over the brain, then one of the things that it does is it will inhibit specifically there because it works as an inhibitor of the NMDA receptor, and it produces acute psychosis. So it produces the same effects that we would see in schizophrenia. And again, there's an interesting sort of anecdote that we see this in adults where this whole circuit is complete, but we actually don't see this in children. So the administration of ketamine in younger individuals, we don't really see that because that circuit that we just examined isn't complete there. So it appears to model or give us a model for hypofrontality. It gives us the elements of the circuit, and it tells us that the feedback and the release of more dopamine, if we don't have enough inhibition and we have less inhibition because of the hypofrontality, it tells us what the circuit is, what the cells are, but it doesn't tell us why this happens. It doesn't tell us how this happens in development. It only tells us that there's something wrong with the circuit. It tells us that we don't have enough of these cortical neurons. We know that there's less of them through magnetic resonance imaging, and we don't have a full mechanism, but it is one potential model system that we could start working on. And again, the whole thing is actually very, very complicated. And one of the things that makes this complicated is, again, we won't go through all of the details. If we look at some of the genetic risks for individuals that actually have schizophrenia, it turns out the immune system and elements of the immune system seem to play a key role in the schizophrenia risk. In fact, the complement component 4 is the strongest risk factor for developing schizophrenia. And why that is really, really unclear. And as we've been looking at throughout this entire course, and we just looked at a model system within the skin, and we looked at the in-V mouse model, it would be awesome to be able to have a good model system that we could go back and examine in a way that is relevant to us as humans. But because of the particulars of how our brain is set up, it would not be really ethical to do any type of experimental modeling on humans. And we don't really have a great animal model system. And so one thing that people have started to do is to use what are known as organoids, or mini organs, to help to see how does that hypofrontality develop, that cortical maldevelopment, the reduction in cortical gray matter. How does that happen? We can't study like infant brains. We would have to remove infant brains. It would be very unethical. We have no way to know if they'll end up being schizophrenic. So it becomes really tricky. And so these organoids actually help us to understand that model system. And these organoids, which are derived from stem cells, sorry, and it's spooling right now, apologies on this. Okay, hopefully it's going to go. And these organoids that can be developed from skin cells are actually the ways in which we were going to link skin to schizophrenia, because we could take skin from both a human that's normal, and we could also take skin cells from an individual that has been identified as having schizophrenia, take those skin cells, re-engineer them back to stem cells that could form all of the different components of the human brain, and see if there is a difference in the development of the brain from these different skin cells. And that, again, is a really important thing. Now, these next few slides that I'm going to skip over really quickly, I'm not going to ask you to know them because there is a difference in the development of skin organoids. But again, for the sake of what you need to know for the final exam, regardless of what it is, final exam or a take-home, you don't need to know these next few slides. Just to indicate that in schizophrenia, when we go through this neurodevelopmental model, it suggests that there are a number of genetic things that contribute and environmental factors that ultimately lead to structural and functional changes that could occur early in development in utero, or they could occur in the early perinatal periods, just after birth, or it could actually occur in childhood, and because of the prodromal periods, it might even occur later on. So there's something going on in development, whether it's genetic or whether it is environmental, that affects the normal development of the layers of the brain, especially within the cortex, and that's what we really want to be able to model. And this theory hypothesizes, this neurodevelopmental model theory hypothesizes that early brain insults actually lead to individuals developing schizophrenia. So something might have happened in utero while an individual's mother was still pregnant, that led to some kind of insult that eventually led to schizophrenia. And this is supported by a lot of findings in these patients that have the schizophrenia and also develop other very, very subtle morphological features that seem to suggest that embryogenesis that leads to malformation of the cortex also leads to other malformations that are detectable on the face or in other parts of their body. It is a very interesting theory. And these brain malformations are supposed to underlie some of the positive symptoms that we've looked at, like the delusions. These are gains. We normally don't have delusions, and they're positive because they're additions to our normal behavior or hallucinations, as well as the negative symptoms. So the affective flattening. We can't really tell if someone is laughing, and we don't laugh as much. Or the lack of motivation, the lack of pleasure, the lack of cognitive symptoms, disorganized speech, cognitive deficits. And all of these things might occur during the first trimester in utero. And again, very unethical to look at normal development in utero as associated with developing a model for schizophrenia. You can imagine the bioethics that might be involved in doing this modeling. So another way to look at this neurodevelopmental model theory is called the minor physical anomalies model or observational model, which represents a broad range of very, very small alterations that occur in the head, the hands, hair, and feet, which also indicate that something happened in morphogenesis during the first or early second trimester, which is the period of time in which your brain is developing. And as a result of those slight intrauterine insults, that your brain or the brain of an individual will have schizophrenic-like alterations. And this has been documented as these minor physical abnormalities or anomalies. And some of these anomalies, again, if you're really interested, is to look at facial asymmetry. So individuals with schizophrenia, there might be an increased incidence of facial asymmetry. They may also have some changes to their orbital landmarks. Their sort of orbital ridges might be a little bit lowered. They may have reduced palatal ridges. They might have changes on the left side of their ear or ear shape. They might have differences in their ears. And these small physical abnormalities seem to be much more common in individuals that would normally have the schizophrenia than in their control counterparts or in non-affected individuals. So again, these are about 110 patients with affective psychoses versus like 158 controls. And we can see that, again, we have quite a difference in some of these different numbers just looking at these minor physical anomalies versus like the controls that are mentioned, and these all tend to be very statistically relevant. Okay, sorry, it seems to be spooling again. So again, although we think that schizophrenia occurs during early adolescence or young childhood, might there be an even earlier insult intrauterine that eventually leads to the behavioral consequences that are seen during adolescence and early adulthood? And there seems to be a lot of evidence that early physical or early insults intrauterine during development might actually lead to not only minor physical anomalies that can be seen on the face or on the hands, etc., but also within the brain. Because if you look, there are inappropriate numbers of neurons in these different cortical layers 2 and 3, in various areas like the hippocampus, as well as in hypoplastic midbrain dopaminergic neurons. And in fact, if we go and look at the dispersion index, which is a ratio of the variance, or how scattered or how widely distributed these neurons are in schizophrenic individuals versus like control, we can actually see that there is indeed a difference. So this dispersion index is quite different in schizophrenic individuals versus control individuals when we look at all of the different layers within the cortex. And if the value is greater than 1, that means that these neurons are very randomly distributed, and we have them in many, many different layers. And if we look at the dispersion index for someone who has schizophrenic individuals, they tend to be more evenly distributed across all of these different layers. And what this really is suggesting, and the take-home message from all of this, is that we measure the minor physical anomalies, but we can also see and detect that the brain structure that we would normally see all 6 layers, we don't have all 6 layers in these schizophrenic individuals. It's not as well spaced out as it should be, and we can determine that through this dispersion index. And again, looking at various layers of the brain, we would be able to detect that we see a difference. And again, the numbers not being important, that we actually see changes in the number of neurons in these different regions of the brains, which suggests that there is an alteration in the normal movement and migration of these neurons that might result in that observation that we had earlier, that the neurons within the cortex are actually, we have less of them in schizophrenic individuals, and this might account for that. So might it occur even earlier? That's sort of what we would be looking at here. These changes in brain structure most likely developed in utero during that first trimester or early second trimester, because if there was a physical insult in the first trimester or second trimester, this is when we have normal neural migration. And we also know that in the second trimester, this is when neurons normally develop connections and synapses. And if in utero there was an insult that resulted in physical anomalies, and we also know that it results in changes in cortical thickness, and it also changes like the structure and the dispersion index in the cortex, that this might indeed account for everything that we've looked at right now. But the key thing is, how can we actually look at all of these different factors? The one thing that we would want to use are these brain organoids. And instead of going through all of the different slides that are left on the slide deck, the most important thing for us to know is that by using brain organoids, and I'll just skip to the slide that is the most important, if we look at the slides, if we look at skin cells, so these are induced pluripotent stem cells that are taken from schizophrenic individuals, or they are taken from controls. And these controls are actually genetically related individuals that do not have schizophrenia, and it doesn't matter if they're male or female. If we look at the induced pluripotent stem cells from schizophrenics versus controls, and we generated mini brains from the control individuals and schizophrenic individuals, this is the normal layering that we would see within the cortex, and this would be the cortical region. And normally we would have stem cells all stuck down here at the bottom of the cortical layers. They would migrate upwards this way, and we get these great sort of neurons that would form the cortex up here at the top. And in schizophrenic organoids that have been developed using the same technique, we see a disordering, and we see that we don't get those nice layers, and these layers aren't really well formed. The dispersion index isn't going to be wide distributions and sort of wide sort of scattering of these cells. They're all over the place. The stem cells are found in layers that we normally wouldn't expect them, so we wouldn't normally see them up here. The neurons aren't all migrating to the layers that they should be. You can see green is everywhere. They're not in the layers that we should see them up here. They're not bright green because we don't see a lot of them. And so these organoids are a way for us to be able to start modeling this. And we do see a disordering in schizophrenia as well as in controls. Now, what should you take away from all of this to get ready for whatever happens on the take-home or the final exam? Without knowing all of the details on how these organoids are developed and formed, which are the other slides that I've skipped over, and again, it would be useful, and I will go ahead and do so and create a distilled slide version for all of you well in advance of the take-home or the final exam. This suggests that taking skin cells from schizophrenics or from normal individuals actually can give us a model system. And based on what we've described earlier in terms of the circuit development and what we've just talked about here, what would that look like if we were to develop organoids, and how would we go back and start examining those different circuits that we talked about earlier? Now, I'm not going to go through that here, but it's something for you to think about, and it's something for you to think about as potentially a short answer question, as I told you that we would start thinking about right before this whole COVID-19 thing took place. These are all the slides that you need to worry about for your final assessment, whatever that form will be. I'm going to ask you to keep connecting every single day on Quirkus, regardless of whether there are updates or not. I'm going to try to be on there more often for you. I'm hoping that you're going to do a couple of things for me. So the survey will be available for all of you over the next few days. Please think about that very carefully. I'd like you to maximize your marks as much as possible. I'd like you to get all of your marks in as much as possible early before your take-home or before your final assessment. Work on your PowerPoints, and if you need any help, please don't hesitate to reach out to myself or to your TAs. I would, if you decide and we get the vote results in, we will automatically and immediately post the marks for your participation as 10 out of 10. And if you can also start submitting your final reflection pieces, I will also start assigning the marks for them. For those of you who have already submitted them, I will post the marks, and all of you did incredibly well. I'm hoping that all of you will continue to do really well on that. And then my next job will just be to keep checking in on you weekly, whether it's in the form of a Zoom sort of open space, and to invite you to a Zoom chat room, or to just start posting like distilled slides that you need to know from all of the different lectures. I will start to do so, and that will be my first priority for you. Now, I'm just going to stop here. I know that we've just been talking for about a full hour and a half straight on. I'm going to check the chat box over here. Please post any questions. If there's anything I've missed or anything that you need to know more information about, please let me know. My job is to make sure that you feel less anxious, that you feel supported, that you know what the expectations are and that they're clear for you, and that you're going to get the best marks that you can in this course. You don't need to worry about any more content being delivered. Concentrate now on finishing all the assignments, not worrying about participation, not worrying about anything else, and just work on your assignments. All right, are there any other questions that I can answer for you now? I'm going to start typing in some of those. Any other questions that I can answer for you? All right. If you do have any questions, please let me know now. If you're good, then we'll just post this. We'll make sure that I start working on the distilled slides. that I start mapping out precisely for you like what you need to get ready. How are you guys for your final exam schedules? Are you hearing back from any of your other courses what your schedule is going to look like? I'd really be curious about that. Sorry, I just missed that. Aaron, can you go back? Can you repost, or Julie, can you repost? No other course. That's surprising. That's very surprising. I'm kind of shocked because that would mean that you're in limbo. Like, wouldn't you be a little bit more anxious? I would love if you would host between now and the final assessment another online chat, of course, where you can run through the distilled slide. Okay, happy to do so. Okay, wow. All right. Yeah, so that part we do not know how the final exam online would be delivered. The dean's office has not been clear on that, and I believe that many courses are waiting to hear back about what the format could look like. There are lots of concerns for me. So students that have accessibility concerns, online testing is not easy for them because there's no way to stop and pause and give them the opportunities um, to be able to read or do other things at the level that they might normally be accommodated for. We don't know about the short answers. All we know is it would be the same time. There would be something in place related to academic integrity, but honestly, we don't have a clear sense right now. How long will the take-home answers have to be? Okay, good question. All right, are there any other questions that I can answer for you while I still have the slide deck available? Slide 33. I can go back and try slide 33. Okay, so Barry, I'm going to try this again, or for anyone who is curious about this, and if there are no other questions, or while I'm doing this, if you can type out any other slides. Otherwise, I think we're going to try slowly wrapping things up. We will be back online next week. Is it okay for all of you next week at this time to be online for about an hour just to check in? It would help me because I just want to know how you're doing. As well as to answer any questions. We'll know the result of the vote, and I'll know how better to be able to help you by that time. So there's one way to help you related to short answers. There's another way to help you related to multiple choice, one-liners, etc. So if you can just let me know on the chat box if you're okay with meeting again online at this time, that would be awesome. But Barry, what is indicated here is that often we have so many neurons. So imagine all of these are neurons, and each of these little dots that you see here is like a little neuron that you have, and it's one cell body of the neuron. And one of the things that we do is we take a look at this whole section, and we can look at this whole section, and one of the things that we know is if neurons are just like randomly distributed, so they're just going anywhere, and they're not actually forming layers, and they're not actually forming clusters in the brain, and we know that that's what they would normally do, then we will actually have a dispersion index that just looks even across like every single layer. And you can see there's layer 6, there's layer 5, there's layer 4, 3, 2, etc. And that's normally how we see the brain. And in a brain that is like randomly distributed, there's no specific layering. It just looks like it's random, and it would have like a value of 1. And we know that if we have clustering, groupings, layering of neurons, that when we look at the entirety of the cortex, so all of this at one time, then one of the things that we would see is we would see clustering of neurons, which is what we have here. So we have a dispersion index saying that these are not randomly distributed, that the dispersion index, which just measures the variance of all of the different cell bodies divided by the mean of all of the reactivity of these neurons that you have in this particular picture, we would actually see that there are actually layers that we can detect, or clustering that we would be able to detect here. In a schizophrenic brain, this number is much lower, meaning that these neurons have actually not migrated, they're not clustering, and we don't get the layering that we would normally have. And so, jumping back to put this in context, when we look at the brain of an organoid that has come from the skin of a control individual or from the skin of someone with schizophrenia, we see great clustering, layering that we would have in a brain organoid from a control individual, but in schizophrenia, it's random. We don't get the layering, and we don't get the clustering that we would have. In fact, it's hard to see like a single layer of brightness, which we have here in the schizophrenic brain. And I'm not sure if that adequately explains that to you or not, Barry. I'm hoping it does. And we kind of link that slide with this slide here. Is that okay for you? Okay, so everyone, I think that's it for today. Thank you so much. Please stay healthy. Please look after your families, individuals in your community around you. I will have the survey available to you, so we had to have had the class first, and then the voting can occur so that you're aware of what you're voting for. And the vote is going to be keep everything as is. The final exam will still be worth 35%. It will still be something that we will follow initially. And your final exam will be online in a manner that we don't know yet at this point. Or the other is to have flexibility around a take-home assignment. That take-home assignment, if you do better, it will be worth 35%. If you don't do as well on that 48-hour assignment, 4 questions, it will be sort of worth less. I would consider it as an open book. The advantages for me would be, from my point of view, is that you would be able to have an open book. It would be worth a little bit less, and you'd have flexibility. In addition to that, we would post to reduce your anxiety, your participation mark. We would just give you 10 out of 10 straight away. We would also be flexible around some of the other deadlines because we know they're all kind of bunching up for you. We would also give you that 48 hours so that you don't have to stress out. It is doable, by the way, completely doable in about an hour and a half to 2 hours. And that's if you're like going back and editing and doing everything else. We want to give you the full 48 hours in case you have other things that you need to study for ahead of time. Regardless of what your decision is, I will prepare a distilled slide version for you, but it will be a little bit different depending on what the outcome of that vote is. I will come back next week at this point in time to just check in on you, see how you're doing, and to see what kind of resources I can supply for you. So at this point, I'm just going to say good night. I hope that wherever you are, you're staying safe. You are really important to all of us. Please take care of yourselves. Check in with me if anything is bothering you, if I can help you in other courses. If you're really still anxious about things in this course, don't hesitate to reach out. Wish your parents well for me, and I hope to see you all again here online at this time. It won't be a long session, and we'll just do a check-in plus give you updates that are important for you. Be well, everyone. Good night, and please take care.